Welcome again, everyone, to PXE International's webinar, PXE, Can Diet Help? I am Christine Vokey, Director of Education and Information for PXE International, and I thank you all for attending today. I want to let you know how you can ask questions during the webinar. There is a question pane in the control panel on the right side of your screen. You can enlarge that pane, type your question into the question box, and we will present it to Dr. Uitto during his talk. And if you're having any technical difficulties, you can also type questions about that into the question pane, and we'll do our best to help you. So I think we're ready to begin. I'd like to introduce today Dr. Jouni Uitto. We are very honored to have him with us today to present this talk on PXE and whether diet can help. Dr. Uitto has been a professor of dermatology and cutaneous biology, biochemistry, and molecular biology, and chair of the Department of Dermatology and Cutaneous Biology at Jefferson Medical College in Philadelphia since 1986. He's also director of the Jefferson Institute of Molecular Medicine at Thomas Jefferson University. He received his MD and PhD degrees from the University of Helsinki, Finland, and completed his residency training in dermatology at Washington University School of Medicine in St. Louis. Dr. Uitto is internationally recognized for his research on connective tissue biology and molecular genetics in relation to cutaneous diseases. The overall goal of this research is to understand the underlying defects in collagen, elastin, and the basement membrane zone, which would explain the clinical signs and symptoms of various connective tissue disorders. One of those conditions is pseudoxanthoma elasticum. Dr. Uitto's group is part of a consortium sponsored by PXE International, first characterized the chromosomal locus for PXE, and then identified the mutated gene as ABCC6. His group has also developed a transgenic mouse, which is a mouse that develops the features of human PXE as it grows. This mouse model is currently being used for detailed studies of the mechanisms that lead to the mineralization in PXE, and also for testing of potential new treatments for PXE. Dr. Uitto's publications include over 600 articles in peer-reviewed journals, and he's received numerous national and international awards, and one of those awards is the 2010 PXE International Research Award for his prolific and comprehensive contributions to the understanding of PXE. And we are very glad to have you here, Dr. Uitto, and with that, I will turn it over to you.
Well, thank you very much, Chris. With that extensive introduction, maybe we have 5 minutes left to talk about PXE, can diet help? Anyway, good afternoon, everybody. This is Jouni Uitto from Philadelphia, the city of brotherly love. And I thought to spend half an hour with you talking about diet and how that might modify PXE phenotype. So just before going to actually diet, let me remind you a couple of things, a couple of facets of PXE. And one of them is that PXE actually can be a diagnostic challenge to physicians for a number of reasons. First of all, it's not a single kind of disease, but there are different forms of PXE, different severity, and so it's a wide spectrum of clinical manifestations. And specifically, there are quite a lot of heterogeneity in terms of what tissues, what organs are affected, how severe the disease, and it can be either interfamilial, in other words, between different families, but also within one family we see quite a lot of variability, suggesting that it's not just a single gene disorder, but there are a number of modifying factors, both genetic and perhaps environmental, such as diet. One facet also which complicates diagnosis is that PXE is late onset. In fact, the diagnosis is usually made in teens, but in some individuals it's not evident until in 20s or 30s. And there are a number of conditions that can mimic PXE in terms of skin findings. So overall, it can be a difficult challenge for general practitioner. Now, the clinical features, the key here in PXE is it's a multi-system disorder, and primarily the manifestations are either in the skin or in the eyes or in the cardiovascular system or in all of them. But there's quite a lot of variability in that some families, one of these three organ system manifestations can be predominant. For example, in some families, it's mostly skin problem, less with the eyes and less with cardiovascular, while in some families, the problems perhaps are mostly in the eyes and very little skin manifestations at all. Now, this is just, I'm sure there are some PXE patients listening, so this is kind of obvious to you. But the characteristic skin finding, the early manifestation of PXE is this kind of little yellowish papule, which are in certain predilection sites. They are usually on the side of the neck or in the antecubital fossae, in the arms or legs, or can be in periumbilical area. But this is the early sign, and then these individual papules start to coalesce. They form these larger plaques, and the skin starts becoming more leathery. It loses its elasticity, and as an end result, we have this kind of leather-like appearance on the predilection side, but can be extending to other parts of the body, and in some cases, quite extensive cosmetic problem primarily. Now, if you take a biopsy from this part of the skin, the affected part here, and stain the skin biopsy. So here's the skin biopsy. This is the outside world up on the top. This is the top layer of the skin. And in the middle part of the skin, when you do specific stains, in this kind of picture, you look into elastin stains. Elastin is a component of the skin, but here there is a clearly accumulation. There's too much of elastin, and it's not normal elastin. This part is sort of normal. You see these narrow, wavy lines. This is clearly pleomorphic, abnormal elastotic structures. And what is characteristic for these abnormal elastic structures is that they become calcified, mineralized. There is a deposition of calcium and phosphate as shown by this special stain. So it's clearly a mineralization disorder. And that is key for the rest of the talk when I talk about the diet here. Now, as Chris mentioned, our group was part of the consortium, which consisted of 4 different laboratories in the United States. And this was in late 90s. We tried to find the gene that harbors mutations in different forms of PXE. And this consortium was put together and helped by PXE International. And PXE International was also responsible for getting the patients' blood samples so that we could identify DNA and find where the mutations are in human chromosomes. And after considerable amount of work, we found there is a critical region in the short arm of the human chromosome 16, which contained the gene that is responsible for PXE. And then further work was needed, first computer work and then sequencing work needed to find what those changes are. And we found, together with 3 other groups pretty much simultaneously publishing, that ABCC6 is the gene that contains mutations responsible for PXE manifestations. Now, ABCC6, what is it? It's ATP binding cassette family 6, member 6. That's the ABCC6 stands for. And this gene encodes this protein, which is a transmembrane protein. So you have here the cell membrane. And this protein kind of zigzags between the inside and outside milieu, and there are these different domains which are critical for the protein function, including these nucleotide binding folds. They bind ATP, which then gets hydrolyzed by this protein, and the energy that's derived from the hydrolysis allows this protein to serve as a pump. It's a transmembrane pump, which pumps something from inside of the cells to the outside to the circulation. Now, one of the early kind of surprises was that this particular protein, ABCC6, is mostly present in the liver and not in those tissues that are affected in PXE, namely skin or eyes or the cardiovascular system, but in the liver. So we proposed this hypothesis, so-called metabolic hypothesis, which basically suggests that in normal situation, in a healthy individual, this pump, which is depicted here, hope you can see it, pumps something out from the liver cells to the blood vessels, to the circulation. And the presence of these molecules, which we still don't know what they are, normally prevent mineralization in tissues, in peripheral tissues. So now, if the pump does not work, we do not get those molecules into the blood, and therefore, in the absence of these anti-mineralization factors, we start getting calcium deposits, mineralization in the eyes, in the blood vessels, in the kidney, and in the skin, as shown histopathologically here on the left-hand panel. So, we still don't know what's happening. What exactly is the molecule that is missing in PXE here, and what are the detailed mechanisms that lead from the mutated protein or mutated gene to the mineralization? So look into this kind of situation more in detail under carefully controlled conditions. About 7 years ago, we developed a mouse model which mimics PXE. And basically, it's you inactivate the mouse ABCC6 gene. It's in mouse in a different chromosome, chromosome 7, but there are all these technologies that you knock out this ABCC6 gene, and then they look at these knockout mice, and they actually recapitulate the features, the genetics, and the histopathology, and the electron microscopic features of PXE. And most importantly, they show exactly similar kind of mineralization as the humans in their eyes and blood vessels and the skin. And again, like in humans, in mouse it's late onset, 4 to 5 weeks of age of the mice, which in fact interestingly corresponds to the human age of about 15, 16. So this is a very good model for PXE. And this just shows actually the mineralization. So this is a knockout mouse, ABCC6 minus. So it doesn't have functional ABCC6 gene in this mouse. And you look at the blood tissues here. Here is arterial blood vessel. You see the special stains show mineral deposits in the blood vessels. You see some in the eye here, these brownish specks. You can find in the kidney. You can find in the skin. But interestingly, we found early on that there's a vibrissae, in other words, whiskers of the mice. They have a capsule, and that becomes actually mineralized very early on. In fact, if you look, here is the mouse, and if you biopsy this muzzle skin here, which the whiskers are sticking out, and you take a biopsy of this, look by histopathology, and here's the whisker hair in the middle. Surrounding that hair is a blood vessel, so-called sinus. And then outside of the blood vessel is a capsule. This is a connective tissue capsule, and this is the capsule that becomes mineralized or calcified in the mice, as shown here, for example, this dark brown color that represents the mineral deposits. And this is an early biomarker of the mineralization process. So we have shown that you can actually look into just this, take a biopsy of the mice, and it tells how much mineralization there is in the internal organs or in the rest of the skin of these mice, or to that matter, in the eyes. So it's a biomarker, and we can actually show that this biomarker is progressive. So that here is, for example, is a normal 6-month-old mouse. This is a normal mouse, has normal ABCC6 gene, and here on the right are knockout mice, ABCC6 minus mice, or the PXE mice. And if you now look at 6 weeks of age, you start getting mineralization, which is not evident in the control mice at all. You start getting mineralization. In 6 months, there is more mineralization. And here, later, there is even more mineralization. So you can quantitate this mineralization by 2 ways. One, you can take a biopsy and measure the actual amount of calcium and phosphate here, and it tells you how much there is deposits, mineral deposits. Or you can quantify by computerized morphometric analysis the amount of mineral there is. So we have a handle to measure the severity and the progression of PXE in these mice, just looking into this mineralization of the vibrissae. Now, and actually this just shows that we looked what kind of minerals there are in those deposits, and this is kind of complex machinery, but it basically shows that there is lots of calcium and lots of phosphate. And if you look in their localization, so here is calcium and phosphate, and you merge these images, they are together. So it's a calcium phosphate complexes, so-called hydroxyapatite, which is basically the same mineral that is in the bone. So very strong kind of mineral deposits in the tissues of these mice, and by inference in PXE patients' tissues as well. So now, question: just a second. Okay, can diet modify the phenotype of PXE? In other words, does diet, can it make PXE worse, or can it help PXE? Why do we ask this question? Well, there were early retrospective studies. These 2 studies, Rennie et al. And Nelsner et al. In mid-80s, where these scientists and physicians went and asked PXE people whether they, what was their diet when they grew up. Basically, they asked if there was lots of dairy products, thinking that taking too much calcium or too much phosphate, which is found in dairy products, that would make their disease worse. And these retrospective studies actually suggested that if you had a history of extensive intake of dairy products in childhood or early adolescence, you could develop a more severe disease. But of course, this was sort of uncontrolled in the sense that you just went to ask people what they ate when they were youngsters, and I doubt that many of us can remember exactly what we had when we were young. There was also more recently, 2005, another study, which was a small group of patients with PXE were fed oral phosphate binders. And these are molecules that bind phosphate and lowers the plasma phosphate levels. And these patients showed actually improvement of their skin lesions, so less involvement. But this was a relatively minor pilot study. I think it was 6 individuals in the study, relatively short follow-up, 1 year. But it suggested that perhaps there is something. So we have now studied more in detail the effects of diet on the mineralization in our mice. And this PXE mice allows us now to study mineralization and the various factors under very well-controlled experimental conditions, because these mice eat the same food, they are in the same facilities, they have the same air, they have the same cycle of the light and dark, same water, etc. So we have a very well-controlled system to look into the effects of various factors. And what I'm going to tell you now is this paper, really, this was published 2009. It was it was the first, in fact, of a series of papers where we've been able to demonstrate that if you increase the dietary magnesium in the food of these mice do not develop mineralization as we see in the mice that are in normal diet. And this is just actually the experimental diet. So we systematically tested different kinds of minerals, for example, and we could show that feeding the mice extra calcium, that did not do anything to the mineralization. Low calcium, high calcium diet, same thing. But here we have the high magnesium group, this group C, which we compared to the control group. And they had exactly the same diet, except these C group mice got more magnesium. In fact, the magnesium is 5 times higher than in the control mice group. So these mice get this magnesium-enriched diet, and then 12 weeks later, we actually go and look if they got mineralization, again using this biopsy from the muzzle skin, the Vibrise assay. And here are, these are control mice. They don't develop any mineralization at all, normal mice. Here are mice, these are all PXE mice, and these mice got normal diet, which have the lower amount of magnesium, and you see there is mineralization. These darkened areas, that's mineral deposits in this staining. Now, if we go to those mice that had a high magnesium diet, there's absolutely no evidence of mineralization. And in fact, I think the next slide shows this more closely. Yes, here's the control diet. You see the mineral deposits. Here's the high magnesium diet. No mineralization at all. So increasing the magnesium in the mouse diet prevents the mineralization to take place in these mice. And we can actually show that by quantitatively by this computerized morphometric analysis. Again, here is our wild type normal mice or normal diet, no mineralization at all. Here is our knockout mice or normal diet, so you get significant amount of mineralization. But if we then look these knockout mice that got high magnesium containing diet, their level of mineralization is exactly the same as in the normal. So again, the high magnesium prevents the mineralization to take place. Now, so we wondered where this extra calcium from those mineral deposits then go. If it doesn't go into tissues, where does it go? So we looked into the urine of these mice. And here is a controlled diet. This is a little busy slide, but try to see these columns that are highlighted with red. And you see here is a mice. These are all knockout mice. These were on controlled diet. And this is the magnesium urinary level in normal mice, calcium milligrams per deciliter. It's about 9 value here. And we have a whole bunch of mice in each group. And then we look those that had actually the high magnesium containing diet. And you see their value is 132. So it's about 15 times higher urinary output of calcium. So that excess calcium goes to the urine. And in fact, what we think happens, that the magnesium replaces calcium in calcium phosphate deposits. And the calcium just gets excreted in the urine. But because magnesium phosphate is less likely to precipitate, it's more soluble than calcium phosphate, we do not get the mineral deposits in these mice. Now, course, you wonder if you drive the calcium out of the system, do you get osteoporosis? Do you drive the calcium from the bones? So actually, we looked into those mice by micro-CT, that stands for computed tomography. So it's like a CAT scan, a little bit like X-ray, where you see the bones of the mice. And here are all these are normal mice in normal control. These are knockout mice on the high magnesium diet, and these are knockout mice. These are knockout mice in controlled diet, and these are those that got a high magnesium-containing diet. And there is absolutely no difference in density of the bones when you do multiple mice. And in fact, we measured the total calcium content of those bones, and there's no difference. So we do not drive the calcium out of the bones. So therefore, it's just the excess calcium that is absorbed from the diet does not precipitate in the tissues, which suggests safety to this magnesium treatment. Now, actually, we did a reverse experiment. This is just hot off the press. It was published in Experimental Dermatology probably about 3 weeks ago, 3-4 weeks ago, and we showed the opposite side. If we restrict the dietary magnesium, in other words, we put very low magnesium in the diet of these mice, they get much more mineralization. In fact, the mineralization process gets accelerated in this PXE mouse model. And I just showed the data here. These are histopathologic slides. Look at the right-hand column. Perhaps this is the easiest. So this is, again, those Vibrissae assays stained for calcium. And here, the top panel, this is wild-type mice, normal mice in high or low magnesium diet doesn't get any mineralization at all because they are wild-type. But if we take knockout mice and put them on the control diet, at 2 months there is no mineralization, but if we put the low magnesium, they start getting lots of mineralization here. And if we wait until 6 months here, knockout mice, control diet, there is significant mineralization, but if we put these mice on a low magnesium diet, there is much more mineralization, as you can see here. And again, we can just quantitate this by showing that if we have knockout mice on controlled diet, 2 months, there is some mineralization, but if we put then these knockout mice to the low magnesium diet, there is much more mineralization. So clearly, the magnesium content of the diet can change the mineralization degree in this mouse model of PXE. So our findings suggest that mineralization process in PXE can be counteracted by a high magnesium in the diet in this animal model. But of course, we need to really study the role of calcium and phosphate and magnesium more clearly in humans. Is it applicable, our findings in the animal model, is it applicable also to humans? And this brings me to a little advertising section here. As some of you may know, there is a clinical trial being planned and soon to be initiated. And in fact, this is run by Dr. Mark Leopold in Mount Sinai School of Medicine. And based on our animal studies, he now poses the question, does increasing dietary magnesium prevent skin and eye worsening of the skin and eye involvement in PXE patients? So he's soliciting PXE patients to come forward. It's going to be a 1-year study. He gives pills which contain different amounts of magnesium. 1-year study, follow up at the changes in the skin and eyes, and hopefully we know the answer whether magnesium is going to be helpful in humans or not. And those who might be interested, they are recruiting patients to enroll. The study is being planned to start sometimes at the first of the year, perhaps in January, and they are looking for 40 volunteers who get compensated for their travel and cost a little bit, get free medicine, etc. And if you are interested, that contact Dr. Abby Moore. Dr. Moore, Abby at PXE International, and here's amore@pxe.org, and the phone number, I think it's the PXE International office number, 202-362-9599, and ask for extension 252, and Abby will be happy to talk to you. So that's a little advertising for this clinical trial that hopefully gives us an answer whether magnesium is going to be good for treatment of patients with PXE. Now, actually, talking about the magnesium, so if you look at the how much magnesium should people take? Well, there is a Food and Drug Administration has a recommended daily dose of magnesium of up to 420 milligrams for an adult person per day. Now, it's not very clear how this was derived quite a long time ago, but it was sort of based on looking what people eat and how much magnesium you get in that food. And in this context, you should know that vegetables, especially green vegetables, such as spinach, is rich in magnesium, but so is tomato and pumpkin seeds. You can probably go to the websites and look specific foods, how much magnesium they have. Grains, especially oat bran, barley, and buckwheat flour, they are relatively high in magnesium. And we know that magnesium is pretty innocuous. So, for example, doubling this amount would not give you any noticeable side effects. If you go much higher concentrations, you may start getting some stomach upset and gastrointestinal problems. But that's what is recommended now, and we'll see what the study will show. As a sort of recommendation overall, healthy lifestyles, exercise is very important, especially those who have vascular problems, and definitely quit smoking. That is a major problem, giving you problems with your blood vessels in the lower extremities, the intermittent claudication problem. So that's about magnesium. Let me just briefly tell you another kind of related, not really diet-related, but perhaps drug-related story, a couple of slides. And this is unpublished story, which was first time, in fact, reported by Kwali Lee. Lily is working with me here at Jefferson on this project. And Lily presented this study for the first time in the PXE International Research Conference that was last week, Monday, Tuesday in Bethesda. So those who were not there, I'll give you a little bit recap here. So basically, we looked into effects of warfarin, and does that change the ectopic mineralization in PXE mice? And really, just to remind you again, the Pseudoxanthoma elasticum, the disease is really a mineralization disease. That's where the problems come. You get too much calcium and phosphate into your tissues, in the skin, eyes, and in the vascular system. And of course, there is the underlying mutations, but how this causes demineralization is not clear. But if we can prevent this mineralization or make it worse, that makes the PXE different. So warfarin, it's actually a commonly used anticoagulant, which is associated with mineralization of the blood vessels in patients that are on warfarin use. So this has also the mineralization as a side effect. And actually, in a experimental model, giving food to rats that contains warfarin makes lots of mineralization to take place. So basically, we ask the question, does warfarin accelerate the ectopic tissue mineralization in PXE? And if it does, that might have some clinical consequences. So what we did, we placed our PXE mice on diet that contained warfarin. And here's actually the summary of our results. Now here's first of all control mice, normal mice. And we gave them controlled diet. Of course, they don't get any mineralization in any of the tissues, other than the kidneys, heart, or etc. And if we give these normal mice a diet which contains warfarin, we get a very little mineralization, just in viprice, 1 mouse out of 9. But if we have PXE mice and give them controlled diet, obviously they get some mineralization in 100% of viprice, but less so in kidneys, heart, etc. But if we put those PXE mice now on warfarin-containing diet, all mice get lots of mineralization. And we can actually show that by histopathology. Let me just pick up here a couple of, so the C is normal diet, D is warfarin-containing diet. Here in the viprice, warfarin certainly increases the mineralization in the viprice. Certainly in the aorta, you start getting much more mineralization in the eye, in the heart, and particularly in the kidney here, as you can see. So basically, and we can quantitate that again by either calcium measurements or morphometric analysis. So that shows here the C is PXE mice on controlled diet here, and this D is PXE mice on warfarin-containing diet, so you get much more mineralization. Now, so we showed that warfarin accelerates the ectopic mineralization in our PXE mouse model, but the question is, should the patients with PXE take warfarin? That's a good question. So to get some answers, we actually have established a warfarin survey together with PXE International. And you may or may not have gotten a email from PXE International to the patients that are in PXE International Data Bank. And basically the survey asks that, are you currently or have you previously been treated with anticoagulants? And if you say yes, the follow-up question is which one, how long, and did your health change while you were on the anticoagulant? Now, we have to be careful in the sense that there are other anticoagulants besides warfarin being used for coagulation problems, and therefore we specify here the warfarin survey, how long you have taken warfarin, and did that change. Now, if you already replied to our email, and I think we got well over 500 replies, so thank you to you. But if you have not replied and you will get another email, please reply. And again, if you have questions, contact Abby Moore at PXE Internal. Here's again the contact info. That's amoore@pxe.org, 202-362-9599, and extension 252 at Dr. Moore's office. So that's about I wanted to tell you this afternoon. Just want to acknowledge Lily Lee and Jia Jiang, who are my younger associates, I might add, here at Jefferson Medical College, who have done all this animal work before. We're very thankful to PXE International for their cooperation and support over the years, particularly Sharon Terry and more recently Abby Moore. And just want to acknowledge, of course, these studies are supported by the taxpayers, National Institutes of Health, National Institute of Arthritis and Musculoskeletal and Skin Diseases. So I want to thank you for your attention and just point out that there is a ongoing series of these webinars. You get info on, again, the PXE International office, and you can access the past webinars through pxe.org past webinar. So thank you very much, and I'll be happy to answer any questions if anybody might have.
Yes, we have a list of questions. Thank you very much for that interesting presentation. I'm going to go back to the mouse. There was some question about the tissues that you look at in the mouse for mineralization. Are they completely parallel to the human tissues that mineralize? And do you look at that in all of those tissues in your mouse experiments?
Yes, we actually, originally when we characterized this mouse, we did a complete necropsy. And this was done in collaboration with Dr. John Sundberg at Jackson Laboratory in Bar Harbor, Maine. John is the world premier mouse pathologist, so he did a very thorough work, looked into every single tissue in these mice. And what is evident that there is a widespread mineralization, similar to humans. There are some human necropsy data, autopsy data, which actually show that there is widespread mineralization. For example, lungs can get mineralization, testes have mineralization, etc. But the clinical findings are primarily in the skin, eyes, and the cardiovascular system. And for example, there is no evidence of the lung mineralization causing any lung problems in these patients.
Thank you. And a related question about the mouse. Does it matter when the mice receives magnesium in their life cycle? So, for example, if they take magnesium later in life, does it still prevent mineralization, or does it ever reverse the mineralization?
Very good question. Very good question. We, in fact, I didn't present the data, but we examined first. What I presented is the prevention of mineralization. So, in other words, here we placed the mice to this special diet at the age of about 4 weeks when they are weaned from the mother's milk and they are going on their regular diet. So that's when we change them to diet. Now, we have done the experiment where we took mice, let's say 6-month-old mice that already have lots of mineralization, and we put them on the high magnesium diet, and then looked another 3 months later whether the mineralization disappeared or not. And the answer is no. It does not seem to reverse the existing mineral deposits. So it stops the mineralization becoming worse. So it's kind of stabilizes the issue but does not revert the findings. So if it turns out that magnesium is going to be useful for humans in as the data may emerge from Dr. Lebwohl's studies, then it appears that the treatment should be started as soon as the diagnosis has been made in order to get the maximum benefit of the magnesium treatment.
Thank you. What happens, can you comment on magnesium and whether it, if you take too much, is it toxic? What happens if you take too much? And should people with PXE start, consuming high amounts of magnesium right now?
The answer to the latter part, no. People should not start because we do not know whether magnesium is going to be helpful in humans or not. And I think everybody should wait a year. When Dr. Lebwohl studies start in January, we know a year from January whether it's going to be helpful or not. And therefore, widespread kind of use of magnesium probably is not advisable. The side effects, they are relatively minor, as I mentioned. Doubling the FDA daily dose recommendation to about 900 probably doesn't give any side effects at all. Very much higher concentrations start getting gastrointestinal problems, and in a very high concentrations you can start getting some heart problems, which are evident because magnesium is used in certain clinical situations on a short-term basis, and they are much higher concentrations than we are talking here. So the answer is probably don't start now. Let's get the facts out first.
Thank you for that. And one side effect of PXE if you're taking, I'm sorry, of magnesium if you're taking too much is diarrhea. So I think you excrete it that way.
Yeah.
Does PXE affect the human blood lab results? You know, any of the lab results you'd look at in a normal blood test? Not, I don't mean a genetic test, but you know, the kind your physician would look at, your cholesterol, your white blood cells, whatnot.
No. Yes, normal routine laboratory tests, they seem to be pretty much on the normal range. And specifically, there is no diagnostic marker or no biomarker to follow the activity of PXE, just taking a blood test at this point. There has been some discussion, some question whether ABCC6 mutations might be associated with hypercholesterolemia, or too much lipids in the blood, but the data are not very clear. There's quite a lot of overlap, and just a routine laboratory test in a single patient is not going to be giving any abnormal values.
Okay. Another question with regard to magnesium. Did you look at the differing effects of different amounts of magnesium in the diet? So you had a 5 times normal diet, but what about 2 or 3 times?
Yeah, we originally looked in fact, and we ended up with the 5 because it gave a clear-cut results. I think we looked into 2.5-fold just in the middle, and it was helpful, but not as dramatic as the 5-fold, and that's why the data are presented this way.
And I think that the clinical trial is going to be giving about double the average daily dose in that trial.
Yeah, I believe Dr. Lebwohl mentioned he was going back and forth with the Food and Drug Administration as to what is the allowable amount. Dr. Lebwohl wanted to have a little higher, but FDA and he settled on 900 milligrams that he told in the last week's research meeting. And basically there are going to be placebo tablets and magnesium-containing tablets. So it's a double-blinded study. So neither Dr. Lebwohl nor the patient knows which treatment he or she is getting, and therefore there is no bias or anticipation. And then Dr. Lebwohl is going to measure carefully the amount of skin being involved, looking into the eyes, see if there are problems, and then taking 1 or 2 small skin biopsy in the middle of the study and at the end of the study, or maybe at the beginning of the study and at the end of the study. And then he will assess whether the treatment, then we break the code and look if the treatment was helpful or if everybody got worse or everybody got better or whatever.
Thank you. We have a comment from one of our people with PXE who is attending the webinar. She says that all the magnesium-containing foods that you mentioned are things that she eats on her vegan diet, and she wants to let us know that chocolate is also high in magnesium. So go for chocolate.
Here we go. Yes, chocolates. Yeah, chocolate
Yes.
and spinach. What a great combo. Yes.
Right. I think that is all the questions we have. Let me check. There is one question about whether this clinical trial can accommodate patients from outside of the U.S.
That I do not know. I think the PXE International website can address that. In fact, yeah, go to PXE International website and they have description of this trial. In fact, it specifies who is eligible. You have to be over 18 years of age. You have to have some visible skin signs, and you have to have eye problems and willing to stick to the diet through the entire 1-year period, etc. But look into the pxe.org and Abby Moore at PXE International can give. In fact, Abby is doing the initial screening of the patients for this trial. So talk to Abby.
Yes, and I can also let you know that there will be 10 required visits to New York, so that might be hard if you're out of the country, and expenses will only be reimbursed up to $3,000, so that is a limiting factor for anyone coming from abroad. Now, do you have any hypotheses of how PXE signs and symptoms might be reversed using diet or medications?
Well, there are a number of different approaches people are working. The reversal is probably a tough kind of nut to crack, because the mineral deposits, as I showed, they really consist of this hydroxyapatite, which is very insoluble, very stable. It's like bone-like material, and it probably turns over slowly by itself. So if we stop, let's say by magnesium treatment, if we stop the further mineralization, then perhaps there is a kind of dissolution of that material over time, but we don't have any magic bullet at this point to test.
And a lot of people are looking for things, including your lab. You conducted a blood draw at the PXE patient meeting last weekend, and can you describe what that project is about?
Yeah, it's actually, we are exactly looking at finding a biomarker which can tell you how severely, or what's the progression, how active the disease, and that would be very helpful in these drug trials, because as you realize, PXE is very slowly progressive conditions. So to make assessment just by clinical examination of the skin lesions, whether they get better or worse, it probably takes a good year, as Dr. Leibwald is doing, but we want something which can be quicker. Perhaps in a month's time, looking into blood test which can tell whether the PXE is getting better or not, and that would accelerate the whole drug development and drug testing. We are also looking just the routine panels, exactly what you were asking earlier, are the abnormal values, and specifically look into the lipids. Lipids issue more in detail, looking some subfractions of lipids, etc. And in fact, we are very thankful to all those volunteers. I think we got about 80 PXE patients donating blood samples, so we'll keep you posted.
Yes. Now, I have a question that goes back to the survey of calcium intake in youth that you mentioned, the old survey that suggested maybe calcium was the amount of calcium you ate when you were a child affected PXE. So, but you also mentioned that low or high calcium diet now you think do not affect the mineralization. Can you elaborate on those two things?
Yeah, so the earlier studies, they were sort of retrospective questionnaire given to PXE patients, and asking what kind of diet you had when you were young. And if I remember the details correctly, these were adults whose account from their diet while they were children may or may not be accurate. So it clearly wasn't a very kind of accurate study. It was suggestive that the dairy consumption, increased dairy consumption, made their disease worse later in life, but not sure how good that study was, because it should really be retrospective, like this magnesium study. Start looking from today on and see what the modulation is. Now, our mouse studies clearly did not show any difference in calcium. Giving the mice diet that contains 5 times more calcium, or a 80% less calcium, clearly didn't change the mineralization. So at least the mouse studies did not miss, just the calcium plays a role.
Another quick mouse question. Did the mice have any side effects from high magnesium?
We did not ask about their stomach upset, but as far as we know, not really, no.
Okay.
Not that I can pinpoint, no.
Mm-hmm. And then one last question. This is about skin. Do you know of any surgical solutions to improve the look of the skin where there are PXE skin lesions?
Well, if you get very loose and sagging skin, yes, some sort of cosmetic surgery approach might be helpful. Then again, that leaves scars, clearly needs have plastic surgeons who are familiar with this disease to do those things. And even if there's an improvement, and there is a significant improvement, but it tends to be sort of temporary in that perhaps a year later you are back to where you were before, because the skin really does not have those functional elastic fibers that are required of sort of normal elasticity and snapback properties of the skin. Right. And then might add there are some thoughts, no controlled clinical trials yet, to use some lasers, laser resurfacing, and actually theoretically you could use some lasers to get the calcium out of the skin, so that would be something for the future.
Will someone do clinical trials on that before it's presented to the public?
I don't know if there are any clinical trials really going on. This has been something under discussion, and in fact, in our place, our laser surgeons just got couple of new lasers which penetrate deeper in the skin, and we've been talking about looking at those, but no results yet.
Okay. Well, I think that is all for now. You've answered a lot of our questions and given us a lot of interesting information and exciting information for PXEers, I think. I want to thank you again for being with us today. And everyone, you can look at our website for future webinars. This webinar will be posted there within a few weeks under the past webinars. Contact us, especially Abby Moore, if you have any questions about the clinical trial or any questions at all about PXE. If we don't know the answer, we'll get the answer from our medical advisory board, one of whom is Dr. Uitto. So Dr. Uitto, thank you very much for being with us today.
Thank you for having me. Bye-bye now.